So I'm here with Gert to talk about what plant and food research has been working on to advance the plant protein revolution. Plant and Food Research is a New Zealand-owned Crown Institute that provides research and development to add value to crop, vegetable, fruit and food products. And our goal is to lead the science needed in New Zealand to develop high-quality, premium plant products. So later in this presentation, we'll give examples of the science, but I'd first like to start with an overview of our innovation strategy. Okay, so you may recognise this slide. It was shown previously today and also presented last year by our presenter, Jocelyn Eason. And it lays out the core foundations needed for innovation success in this area. It's very much a holistic approach. And it's important for New Zealand to have development across all of these areas. And as you've heard in previous talks, there's a large opportunity for us. The market growth and industry investment and innovation continues to rise for the alternative protein space, and we have an opportunity to capture some of this. And today I'm going to focus in on the high-value plant ingredient manufacturing, and Gert will talk about product development and how plant protein structures can be enhanced using fermentation. Oh dear. Here we go. So the team that I am part of, many of which are here today, have over 90 years experience in protein ingredient development. And we have been working with national and international clients in this space, helping them to commercialise. Part of the requirements we have with these clients is to have confidentiality agreements in place, so there'll be some details I cannot talk about, and I will need to generalise in some places. So this is a simplified summary of what's involved in the development process. We start with evaluation of the potential protein sources. Then we develop processes around post-harvest and the manufacturing steps. And it's very important to build in quality and food safety into this. We use various models to help us with optimization and to track the likelihood of success. This is for environmental, economic, and processing attributes. And finally, we finish with evaluation of our products and making sure that they are meeting the target properties that we want. And I'd like to spend a little bit of time just explaining what this might look like. We're interested in high-value protein ingredients, and there are three key properties that we're trying to target here. Firstly, a good sensory profile is taste, texture, appearance, and flavour. It needs to be attractive to the consumer and the customers. Then there is performance and a functional role in food, and this includes the formation of gels and foams and emulsions. And finally, high nutritional value, which has been discussed in depth today, and this includes a balanced amino acid profile, and potentially incorporating some of the other beneficial parts of plants as well. So here are 8 examples of potential crop sources, and these have come from a white paper published last year, which Plant and Food Research wrote, and it was named Opportunities in Plant-Based Foods, and you can see a link for this on our end slide if you want to have a look. So we have examples of cereal crops, kiwi fruit seed, forage crops, amaranth, canola seed, peas, hemp seed and potato. And all of these examples of potential sources need to be evaluated against a series of questions to challenge them and to make sure they are viable choices. So we start off with how well can they be grown here? And very importantly, do we have an advantage in New Zealand for growing them? And if we do so, what is the environmental impact? What is the yield of protein going to be? And are there uses for all of the other non-protein components? Do we have room in New Zealand in terms of the IP space to develop processes? And finally, are we avoiding commoditized and subsidized crops from overseas, and are we capturing a niche part of the market? So protein is not the major component of these crop sources. Here we have two examples. One is oil seed, such as canola, and we have forage leaf. So in canola, protein is about 22%, and the majority is oil or lipid. And this is already extracted in existing industry as an oil product, and protein can be then extracted from the pressed meal cake. Then we have leaf, and the protein content is about 4%, and the majority that is there is water. And the water and the soluble protein can be pressed away as a juice in the initial phases of manufacturing. So two key points here are we need to find uses for the non-protein components. And secondly, there are differences between the proteins and also what else is there, which has a big impact on how the end product will behave in terms of those three key properties that I talked about. Here we go. So in a previous role, I worked in quality assurance, so I understand the importance of incorporating this into the development process. And for plants, this is important because they come along with compounds which can be harmful to humans at certain concentrations. So we're talking about toxins, anti-nutrients, and allergens, for example. And also, microbes can be introduced from the production environment, which need to be controlled for. So we need to design our process to mitigate against these. And there are two challenges that we are facing in this type of process. And one is that we are trying to concentrate protein, and therefore we may be inadvertently concentrating some of these unwanted compounds if they have not been removed. And also some of the traditional techniques and methods for processing and reducing these, such as heating, may have a negative impact on the three key properties that we're trying to preserve. So it's important to monitor these and design the process to preserve them. So moving on to post-harvest, it's very important how the crop is handled after the harvest point, especially for high moisture crops, as there are enzymatic reactions which can lead to degradation. And it's therefore important to have optimal storage and conditions in place to reduce this. Here is examples of our work with leaf, and we were interested in the recovery of soluble protein from this. And we were studying here the effect of chilled storage over time and also of drying on what the soluble recovery could be. We found for chilled storage that the levels were able to be maintained and were stable. However, we recognise that there is a microbial risk if you do this kind of storage for a long period, obviously. So drying is one way to mitigate against this. However, we found a strong effect, a negative effect on the amount of recoverable soluble protein. So the optimal conditions for this type of crop is to process as quickly after harvest as possible and possibly introduce interventions to reduce microbial loading, such as UV treatment is one possible example. So once we have harvested a crop, it needs to go into a manufacturing process, and this diagram gives some of the principles involved. We have a protein ingredient on the top right there, and along the way we produce various co-products which we need to find uses for, including some bioactive opportunities such as vitamins and polyphenols. We start with a primary separation, and I've already talked about pressing oil and juice, so that's an example here. We also have the need to move into further isolation, concentration, and purification. And there are a wide range of technologies and methods available. And there are trade-offs between them about the yield, their operating costs, and also whether or not they have a negative impact on the quality. So we can screen some of these methods at a lab scale as a starting point, and can also get information about the compositional and nutritional properties. And you can see in some of these pictures the work that we've been doing. On the bottom left is extractions we've been doing with canola meal, and we've trialled different pH conditions, and we're measuring what proteins were in fact extracted, and not only that, what were the functional properties of these and the bioactive properties of these. In the centre we can see the continuation with the leaf work that I talked about in the post-harvest slide. Not only did we see differences in the soluble protein recovery, but in the bottom picture there you can see a difference in colour. So on the left is the chilled material and on the right is the dried, so there's a visual difference there too. And finally on the right is work with purification techniques, and we have absorptive material in that column to remove some of these unwanted compounds. The trial work at lab scale can only give us so much information. We need to move into larger scales to inform what could happen on a commercial scale. So we work at hundreds to thousands of kilograms of material in a pilot plant environment. And here we're gathering information about yields and composition and quality and just general on-hand practical experience with the product. And this is used to build our optimization models and to inform large-scale design. So in those top pictures you can see work we're doing with filtration technologies, both for isolation and for concentration of protein. And then we have a picture of a spray drying process and also a collection of one of our prototype ingredients. So I'd like to finish up my part of this presentation just looking at some of the properties that can be achieved with these protein ingredients. And these are all pictures from our food development lab, showcasing functionality of the various prototypes that we have created. So in that bar, that's a high protein formulated bar, and the protein is performing a role in maintaining the moisture levels throughout that and preventing staling and giving it the soft texture. In the top right, we have a protein gel. And this has obvious applications in the meat analogues and dairy analogues that we've heard about. And in the bottom there we have one of our proteins that's gone through a foaming test, and this has produced a large volume, very stable foam, and has applications in dairy and dessert type products. So I'm going to finish with a short video showcasing another functional property, which is dispersibility in water, which is very important if you want to further process the protein ingredient, or if you're developing products that need to be rehydrated. So on the two outside samples we have commercially available protein isolates, and in the centre we have two of our prototypes, both from the same starting material. And you'll see that in B, the dispersibility is very, very good, and we've targeted this through our processing techniques to achieve this particular attribute.
So today we've seen a range of examples of use of plant proteins in new products, and some of these are really improving. Having said that, current products still are of lower intrinsic quality, and an ongoing challenge remains is how do we create foods that are healthy, that consumers desire and add value to the New Zealand economy. One of the ways that we believe this can be done is through fermentation. So my name is Gert. I work as a scientist for plants and food, and today I'll be talking about a research project that I've been working on for the last year or so. This is very much a work in progress, so please bear with me. And the work that I'll discuss covers two arms of this work. One is around structuring some of these plant proteins, and the other aspect is to use fermentation to further develop the structure and flavor of these foods. So I put up examples. So fermentation is not necessarily new. This has been done in a range of products, and whole food fermentation has been used for a long time. Here are some examples of pickling. The top two products, one kimchi and the other one sauerkraut, two cabbage products that have, after fermentation, have quite different organoleptic properties. They're quite different when you eat them. One of the questions that we asked is how do we extend this fermentation into new products, more complex products, such as yogurts and maybe even cheese? Before I go into more detail, I thought I'd just briefly cover what I mean when I say structure, food structure in its own right. And I've put up 3 examples for to emphasize this, and this is on a moisture content scale. So you've got your very liquid products, your drinks, your semi-wets, which have a moisture content around 40 or 50%. So you can think here of products like your tofu, but also sausages, cheeses, those sorts of products. At the far end, the solids. So examples of that are bread. In here, it is the food structure that is really important to get that right. If you think of a bread that isn't aerated, obviously that eating experience is very different. So food structure to get that right is really important. For us as scientists, this leads to, we want to understand these, we need to understand how these are, what they look like and what, how they work. For this we have a range of tools. Some of them are physical tools like the rheometer, the texture analyzer and microscopy to get some numbers on what that structure looks like, as well as more advanced techniques like NMR or thermographic analysis to understand how free and bound water behaves in this food. None of these techniques are a substitute for sensory testing. It's enormously complex, and ultimately that is where we have to go, but obviously we can't do that for all steps of the way. Cool. So for the products that I highlighted, we don't have an existing product that we can start with. So we have both a challenge and an opportunity. For the products I put out, we sort of see we need 3 components. First being the functional ingredients that Thomas just highlighted and introduced. And so here you can think of a range of protein sources. You can from your legumes to oil seeds. And the work that I show today is using pea protein, and that was chosen mainly on the availability of this product. These foods have lipids, so obviously we want to add that, and again we can choose different lipids depending on what you want to achieve. And lastly, we need some sort of carbohydrate if we work with an isolate at least, and that's mainly to direct the fermentation. We're talking about lactic acid fermentation here. So the structures I'm trying to make and fit all into the gels or a 3-dimensional structure, and it's the proteins that provide the scaffold for this. What you're looking at are 2 different ways of structuring the pea protein. I've chosen these to give an example of how important your structuring method is and what can be achieved. So both are made from the same pea protein and at the same concentration. The example at the top uses gluconodelta-lactone. This is an agent that once added to water slowly acidifies the media. When you do this with pea protein, you end up with a weak gel that is self-supporting, and you can see that in the middle at the top. If we look a bit more detailed at this structure, using confocal laser microscopy, you'll see that this is built up of small coagulates of protein that holds this all together. When you work with this, you might experience that it's quite pasty. To contrast this, we can use techniques like cross-linking. So the example given here is for transglutaminase, and what it does, it covalently cross-links proteins together, which extends the protein chain. This has a marked effect on its water holding capacity and elasticity. So again, we have a self-supporting gel, but as you can see in the image, it's quite different in its behavior. It is, the elasticity really contributes there. So this is where we are. We're looking at these different methods, and the examples I give here are not necessarily good or bad, but they are examples of what we look at. Next will be to incorporate those lipids and we'll combine that with fermentation and hopefully find which ones are more suitable. So fermentation, the breakdown of a substance using bacteria, yeasts, and microorganisms. And if controlled correctly, then this helps with the preservation of foods, and has been used to enhance or improve the properties of a food. There's other benefits that come with fermentation as well. So it has the potential to increase the health potential around digestibility, addresses some of the anti-nutritionals that Thomas raised before, reduce allergenicity, as well as to support to recognise there's other flavours associated with plants, and fermentation might address some of the off flavours in this area. I lost that one. So this brings us to the microbes. We've obviously talked about fermentation. So what we're looking for are starter cultures, so a collection of microorganisms that we can use to direct the fermentation to ensure that we work with a food-safe and consistent product. In the plant protein space, there isn't a product readily available out there that we can isolate this from. So we were looking for a source that was rich in microorganisms that we could use. And we used spontaneous ferments, the jar that I show here, so where we created conditions and directed the fermentation so that we could isolate organisms with the traits that we were after. So the example here, so people might recognize this as a sourdough. We didn't use wheat in this example, we used pea, because based on the assumption that organisms that come from the source material are likely better capable of breaking this down successfully. We did this with different source materials, so we did do combinations of pea and wheat, for example, and you get quite different products out of that. These starters appear to be quite different. So we generated a range of these and isolated a range of bacteria from this that we could use for our products. So next was around the characterization. Obviously, we want to know what we work with. First, we need to identify where do they belong. We obviously want to work with food-safe organisms, and so for that reason we focused our work currently around lactic acid producing bacteria, and that's mainly because of their recognized food safety. Next, we talk about flavor development. These are the volatile compounds that these organisms produce, and individual isolates produce different volatiles. So as an example, some might produce a lot of esters that you might recognize also from fruit, so esters play a key role in that. In the corners is acidification, so we are looking for lactic acid production and acid. We want the organisms to do this rapidly, and this is for food safety and to alter the food environment of what we've created to reduce the chance of pathogens to thrive. Last, as another example, is the proteinases. These are enzymes that break down proteins into smaller peptides. We talk about a high protein food. We need these organisms to live in this media. Inherently, they don't have all building blocks. They need to salvage that from their environment, and this enzyme plays a key role in that. Why do we care? It is this breakdown and the breakdown products that we come to appreciate as when we eat them. It's what gives fermented foods their distinct flavors and properties or what we're looking for. Last, this is not where we are yet, but what really is the next step in this project will be to bring both the structuring and the different isolates that we have together. We do this in the form of concepts or prototypes, so these are not necessarily your final product, but what we want to do is bring these together and see how well do they mimic, or what changes do they invoke in these food products. And so we're quite excited to start doing this work, because obviously this is where we've been leading up to. So yeah, keep an eye out. Hopefully we'll be back next year. Lastly, I'll just, to close up, and I think I'm going over time slightly. So what you've seen today are two aspects of what plants and food has to offer. Thomas introduced a complex area of developing functional ingredients, so talking about different separation and purification techniques to get these ingredients in hand whilst maintaining food safety, nutritional and functional quality. My work is around a new product development and using fermentation to develop these new exciting foods. This is part of a wider capability that plant food has to offer. So from lead cultivars all the way through to vertical integration. So we can help in a range of research questions that you might have. And this is also, we cannot do this alone. We need industry to be successful, bringing these products to market, but also just being successful in the plant protein space. Lastly, I'd just like to draw your attention to the link up on the board. This is the presentation that Jocelyn Eason gave last year. So if you're interested in a bit more of the wider capability of plant and food, please have a look. And lastly, I'd just like to thank you for listening.
Yes, we're going to do some questions. Thank you very much, Thomas and Gert Jan, for that. Some great questions coming up, and people are getting really into this. I'm going to go down to the second one because that's the question I saw first. Can you explain on a practical level how something like peas go through the process to become something like sun-fed chicken without giving away any trade secrets? What are some of the stages in the process for transforming peas?
Sure. A key part of their process is extrusion technology, where they're creating protein fibres from the pea under high heat and shear to create long fibres which give it that texture. Yeah.
Thanks. But don't try this at home, I think, is the underlying message there. It's obviously quite complicated. Again, I don't want to dive too deeply into trade secrets of Sunfed, but is there actually quite a lot of proprietary technology that actually gets you to a particular, some of these particular protein alternatives?
Extrusion has been used successfully for a number of these products. Companies like Sunfed do find a niche. There's the combination of your ingredients and what types of processes, including extrusion, that you might apply that ultimately leads to the success, which is challenging to navigate, and that's why not everyone does it. But if you do it successfully, you get products that are good and interested to consumers. So, yeah, I don't know if we can give specifics, but I don't know to answer.
No, it does help, thank you. But it obviously leads on to another great question, which is now the first one, about the extent we've got infrastructure in New Zealand to be able to develop and commercialise such products. What's your sense of the capability here in the country?
So our team in Palmerston North works at Massey University a lot with the food pilot plant. So that's one resource that we have to access. However, we don't have in New Zealand a dedicated plant protein processing facility at this scale. So we're hoping we can build momentum towards that, and that will help with our development process in New Zealand.
Thanks. And some other, in a sense, related questions there. The work you were showing today, was that market-led or science-led? Were those projects that some individual clients, it sounded as though, had asked you to do?
Yes, so for my part of the presentation, it was very heavily involved with commercial partners, interested in manufacturing ingredients on a large scale.
For my work, it was when I joined and looked into what was done in this area, we recognised that that's an area that is less developed and might provide a unique opportunity for New Zealand. So why we put that forward.
Right, thank you. And next one down is the challenges about scaling up fermentation processes. Beyond breweries. I mean, breweries have got it down pretty neatly.
Fermented products are produced at scale. How challenging that will be for these products, I don't know. We're very much at the start of this road, and do I believe that we can do it at that scale? Yes. What that looks like, that will be part of that journey that we have to take.
Thanks. I think that's a signal. So I'm very sadly going to have to stop at that point. But thank you very much for being at the leading edge of the science of this, and greatly look forward to seeing how the science progresses and what gets commercialised from it. A very big hand, please, for Thomas and Gert.